Hello, this is Ray Enke at James Madison University, and this is DNA Subway Purple Line tutorial number 4. And where we left off last time is we had set our core metrics using data from the alpha rarefaction and data 2 steps to determine our sequencing depth. And so I'll just do a quick refresher there. So we set our sequencing depth for, I think it was 632, and that was a sequencing depth that was not necessarily going to provide the most robust analysis for Purple Line, but it was going to allow us to not drop off any samples. So I would definitely advise to maybe run this step again with a higher sampling depth to see what changes you get. And then in terms of checking, selecting your classifier, you want to make sure that you select this green genes classifier. These are the 16S primers that we used for our analyses. So the core metrics, when that's finished running, that will open up alpha diversity, beta diversity, and taxonomic diversity stops on the Purple Line. So let's talk about these 3 stops. Alpha diversity is going to show us plots that are telling us about the diversity of taxa within each sample. And there's a couple of different algorithms: p-lose evenness, Faith's phylogenetic diversity. I'll just show you p-lose. They're both pretty similar as far as I'm concerned anyway. And so what we have here on the Y is a plot of p-lose evenness, which factors in both species evenness as well as richness, so the number of species within samples as well as a count of the individual taxa. And we can sort based on metadata. So in this case, we might want to look at sex, so the difference between female and male samples. Or within female and male samples, you can also just look by individual samples. So here's all of our individual samples splayed out. So these plots are showing us the sample diversity within each individual sample. So as opposed to within sample diversity for alpha, beta diversity is looking at the diversity between the different samples. And again, there are a couple of different ways you can look at these. I usually use Bray-Curtis or Jacquard, and these are plots. When you look at these are principal coordinate plots, and these are plots that are, I think, often most intuitive for students, for me as well. And so what we have here is each sample is plotted. We're going to select our metadata that we want to sort by. So again, in this case, we're most interested in sexual dimorphic differences between these snake musk samples. So I'm going to select sex, and what you can see here is that we have females in red, males in blue. This is an interactive plot we're seeing in 3 dimensions since we have axes 1, 2, and 3. And what we can see here is that there seems to be some pretty clear sample clustering of male and female samples. So this is pretty exciting to us. There's also a lot of other options you have here. So if you click over here on this axis tab, I'm not a fan of the black background, so I like to swap out the black background for white background. I feel like that's an easier thing to look at. You can also use this shape tab if you want to sort one of your other metadata metrics using shapes. So we have color indicating male, female, but something else, any metadata, any other metadata you have, you can sort by another piece of metadata. So for instance, we had different tanks. So we had tanks 1, 2, and 3 that these samples were coming out of. So we'll call tank 1 spheres, we'll make tank 2 diamonds, and we'll make tank 3 cones, and we'll do the same for, well, there's only 2 tanks for males, but we'll do the same thing. And what we can see now is you can now sort by a couple of different metadata metrics. So we have color indicating male, female, but then we have the shape of these points indicating the tank that they came from. Okay, and then this last type of diversity is the taxonomic diversity, and this stop on the Purple Line gives you these sort of canonical bar plots, which are kind of a hallmark for microbiome analysis. So I'm going to make my screen smaller, but you have this little slider that you can make your bar widths very wide. So what we're looking at here is the relative frequency of taxa from 0 to 100%. And then you can sort this based on, well, let's first go over here and we'll look at our different samples. So now across the bottom, we have our female samples and our male samples. And then we have to pick what level of taxonomy we want to sort by. So this is your, you pick 1 through 7. This is your kingdom, phylum, class, order, family, genera, species. If we just look at level 1, this is kingdom. And as you slide over these bars, you can see that they're 100% bacteria. So that's not surprising. It's actually a good thing. I'd be worried if we saw something else. Let's look at level 2, which will give us phyla. And now you can see we're getting a breakdown of all the different bacterial phyla that are in our samples. And each one of these bars is giving us not only telling us which taxa are in there, but they're giving us a relative frequency. And you can toggle over these bars with your cursor and you get the actual percentage number. You can also go over here to the legend and you can select individual taxa that you want to pull out. And what we're seeing here, even at the phylum level, we're seeing, there seems to be definite dimorphic differences. So this is a finding that we're really excited about for our project, and we're going to look to explore more. Let's go to level 5 and we'll look at the family level. Keeping in mind that these 16S reads, these short 16S reads, are not always going to call your taxa to the species or even genus level. So you do have, this is another area that with your data you have to play around with which level of analysis is going to work the best for you. But yeah, this looks really good too. There's definitely some interesting differences between our male and female samples that we need to go and kind of drill down deeper into. So this, these taxonomic bar plots, as well as the principal coordinate clustering plots, the beta diversity and taxonomic diversity, these are sort of your bread and butter data outputs that you see a lot in microbiome papers. Also alpha diversity, depending on your sampling. So there is one last stop. If you're interested in using this differential abundance step to make a heat map, this is something, I'll be honest, I don't use very much, but you can pick your metadata that you want to group the data by. I selected sex. You have to select your taxonomy. I selected, for the job I already ran, I selected, I think it was 5 family. And what you get out of this is a heat map that looks something like this. So this is a little bit hard to look at, but there may be a utility for this depending on what your analysis is. Okay, well that sort of summarizes all of our DNA subway purple line steps. So hopefully you saw how easy this is to use and how much useful data you can get out of it. So I hope that you will use this for your own analysis.